All right. So, hi, my name is Shane McInally. I'm a postdoctoral researcher at Brandeis University. And today I wanted to share with you my wandering path to becoming a scientist and share some of the interesting scientific problems I've encountered along this little journey. And so to start, I was born and raised in Southern California. And so that might evoke some thoughts about the nice beaches, maybe Disneyland, or some of the cool cities that are found in Southern California. But where I grew up was actually in a small town in the foothills of the San Bernardino Mountains, in a small town called Yucaipa. And when I was a high school student, my priorities were mainly focused on playing ice hockey. Yes, we do have ice hockey in Southern California. Playing music, listening to music, and honestly, really sleeping. So I found science really interesting, but school just wasn't something I took very seriously. And so it was somewhere on the priority list, but it definitely wasn't toward the top. And so my parents had always encouraged me to attend college, but I really didn't know where to begin as a high school student. I didn't really know, when to take the SATs, what really was a GPA, how to apply for college. And somehow I managed to navigate through those obstacles. And I ended up applying to and attending the closest university to where I lived, which was UC Riverside. And so as you can see here, I started off in San Bernardino County, moved right over the border just into Riverside County. And when I got to Riverside, I really learned that I didn't have the tools to succeed because I wasn't taking school seriously early on. I didn't really know how to study. I wasn't a very good writer. I just didn't have the tools to really succeed and do well in the college environment. So it took a lot of work and a number of years for me to actually learn how to, study and how to do well on exams and do well in college. And that's really what I had to do the first few years when I was at UC Riverside. And so while I was there, I was actually learning a lot of science, but it wasn't through my courses that I actually sort of fell in love with science. That actually happened when I started working in this building here. This is the genomics building at UC Riverside, and that's the place where I first joined a lab. And once I got into the lab, I really got exposed to the exciting world of research and really the process of exploration and discovery. And that is really what hooked me in science. And so when I joined this lab, the main technique I was using is something called electrophysiology. So electrophysiology is a technique that allows you to see neurons and their behavior in real time. And so what we would do is we would take fruit flies or other organisms and kind of restrain them in these little plastic tubes and lay them out so that way we could look at these antennae that they have, which is their main sensory organ. And then we could put these flies on these slides on a microscope and probe them with these various electrodes. So that way we can actually look at how these neurons would respond to different stimuli. And so to show you an example of what this would actually look like, as you can see in this video, we have this very thin glass electrode that we then probe into the antenna, and then we can actually see these spikes, which are the neurons actually responding to different stimuli. And so you can see that again here. So we have some large spikes, some smaller spikes, indicating that there's different neurons housed within this little structure of the fly. But what's really cool is then you can expose these animals to different compounds and test how these neurons respond to these different compounds. And so you can see in this video here, we can see our normally firing neuron. And then once it gets exposed to this particular chemical, it actually excites the neuron and evokes like an excitatory response in that neuron. And so what's cool about this technique is you can do this for various different types of insects. And one thing that we focused on were disease-transmitting mosquitoes. And the idea is, can we actually use this technique to identify different repellents or attractants to help try to prevent the spread of disease? And so to test this, what we did is we actually went to Kenya to this institute called the International Center of Insect Physiology and Ecology to conduct semi-field experiments to see how well the repellents and attractants that we identified in the lab could actually work in a field setting. And so getting there was non-trivial. We had to fly first to Nairobi here, the capital of Kenya, then take another flight to another large city called Kisumu, then drive along the edge of Lake Victoria, finally taking a very scary ferry ride across this little bay here, and finally made it to Mbita. But the reason that we went through the effort to go on this really long and somewhat complicated journey was because at ICIPI, they have this really unique research setup where they have these greenhouses that you can see here. And within these greenhouses, they have these different huts that are designed to resemble the structures that people live in the local areas. And within a single greenhouse, they have two sort of huts, one on either end. And so what we did is we developed these traps here that are designed to expel carbon dioxide at the same rate that humans do. And the carbon dioxide is the main attractant for these different types of mosquitoes. And in each of these huts, we would put one of these traps, and then one of the huts we would treat with the compounds that we had identified in the lab, and the other we would treat as our control hut. And then what we would do is take this coffee cup filled with really hungry mosquitoes, release them in this greenhouse, wait overnight, and then see how many mosquitoes were actually able to get in each of these huts. And what was really interesting is we found that the chemicals we found in the lab using electrophysiology were actually able to reduce the number of mosquitoes that could get into the huts by about twofold. And so while this was really interesting, I wasn't really super interested in developing new repellents. But what I found interesting was this sort of change of perspective, thinking about individual health to health on the population level. And how could I help try to affect health for communities rather than just individuals? And so with this in mind, I decided to go to UC Berkeley, and I studied public health with a focus on infectious diseases. And I don't have much time to get into this story, but I ended up spending the summer doing research in India, trying to measure the prevalence of drug resistance in the community there for different bacteria. But the important thing, at least in terms of the story here, is that this actually exposed me to how little we know about these different pathogens, especially those pathogens that affect people that don't live in the Western world. And I was really surprised that there was just a lack of basic information about how those organisms grow, how they live, and how they can actually cause disease. So with that in mind, I decided to get a PhD in microbiology, and I intended to study microbial pathogenesis. How do these actually pathogens cause disease, and how can we help find cures for those? But then I kind of got down a rabbit hole because I read this paper. So this paper really put me on the track to where I am today, and lays out the questions that I'm still trying to answer. And the reason I found this paper so interesting is because it posed questions that I had never thought about, and I had no idea how to answer. And so the first question, the one that really made me put this paper down and just sit there and think, was how do cells, or why do cells care about the size of their organelles? And in my head, I'm going like, wait, what? Cells care about how big the stuff is inside them? I never had thought about that. Why did they care about it? How did they do this? It was a whole new question for me. And so when I was taking courses both in high school and at UC Riverside, this was kind of the way I thought about a cell. This sort of generic cell has a nucleus, it has some mitochondria, it has a Golgi, an endoplasmic reticulum, and maybe some other parts there. But it was just mostly uninteresting, just a list of parts. But what I found out is that cells are much more complicated than this. And so when we look at the inside of a cell, so this is real data from a pancreatic cell, we can see that all of these different colored structures have different shapes and sizes, and the inside of a cell is actually a really complicated environment. What I also learned is that organelles and parts within cells are really highly dynamic structures. And so this is one of my favorite videos because it shows us the endoplasmic reticulum in purple, a cytoskeletal filament called the microtubule in yellow, and another organelle called the lysosome, all in different colors here. But what you can see is that they're all highly dynamic. They're moving around, they have different shapes, different sizes, they're fusing, they're dividing, they're doing all sorts of different behaviors. And these are things that I had never really thought of before. And so it's really impressive to me that these cells can control the size of these parts, but they're also really highly dynamic. So how is it that cells can actually do this? And so the main thing that I focus on is the cytoskeletal system found in different cells. And so in this image, you can see not only is it very beautiful, you can see that there's different cytoskeletal filaments labeled in either green or this sort of blue color, the cyan color. And hopefully what you can appreciate is that even though you see a lot of different blue and a lot of different green in different types of organisms, they all kind of look a little bit differently. And somehow these cells are able to use these really common, highly conserved molecules to build very specific structures to help solve specific biological problems that these organisms face. And so what my research focuses on now is how are cells able to do this? How are they able to control the dynamics that lead to the assembly of these different structures to make them specific sizes, so that way they can conquer these different problems or issues that these cells face. And so one way we do that now is through using really simple computational and mathematical models. And so there might be a lot on here. There's actually some graphs, but what we can really think about is just treating these structures as Legos. And so you can see here we have a couple little Legos. And this is exactly how these cytoskeletal filaments work. They simply just add more Legos to make the structure bigger. And sometimes they remove some of these Legos to make them smaller. And so in this situation, kind of what we have drawn here with this unregulated filament, the rate of addition represented by this K plus and the rate of removal, K minus, both, they don't know anything about each other. They don't have any information about how big the structure is. They just kind of add stuff and remove stuff stochastically. And when this happens in a cell, you actually don't get very well controlled structures. What you see here is you just end up with a lot of very, very small structures. So this is the probability of having a structure of some size. And as you can see, very often that structure is going to be less than 5. But what we can do is invent a very simple rule. And this is a rule that cells use very commonly, where now, instead of having this rate of addition just be completely independent, it's actually going to depend on how many Lego pieces I have. So here I have 7 different Lego pieces. So when I have a lot of Lego pieces that aren't in the filament, I'm much more likely to add more Lego pieces. That is until I run out. And once you run out of Lego pieces, there's no more to add. All that can happen is they can fall off. And so that's what's called the limiting pool model, where essentially the size of your structure just depends on how much stuff you have in the cell. And importantly, we don't want to forget about this process of disassembly. When these two rates meet, you end up with a state of equilibrium where you're just as likely to add a piece as you are to remove a piece. And ultimately, this is a strategy that cells use to actually build structures that have sufficient size to solve some of these different types of biological problems they have. And so the research I do now actually kind of lies right at this interface between the theoretical physical modeling and quantitative experiments that we do in the lab. And just to finish off, I think what some of the more important messages are, instead of focusing just on size control, which I could talk about forever, is really thinking about how, if you look at the career path that I took, and many different scientists that you might talk to, very rarely are these career paths linear. You kind of hop around and do a bunch of different things. And I think that's very fun because you get to explore different realms of science. You get to do different things and discover new things. I mean, that's really why I'm here today doing this type of work, is because I get to discover things and see things for the first time that nobody else has seen. The other thing I want to impress upon you is just say yes to these different opportunities. Many times I found myself in a situation where I felt unqualified or just unable to do some of these things, but just say yes. Don't self-select and say, well, I don't think I quite fit. Just go for it. The worst they can say is no, and just get out there and try to do different things. And lastly, I think science is just a really excellent avenue for exploration. It's a very fun, exciting thing to do. And so I encourage you to stick with it. I think it's very cool. And so with that, I would take any questions.